Yeah, so I'm going to change gears a little bit from what we were talking about previously. So I'm not going to talk about the center so much as what I want to do is kind of give everybody just a little impression about how we think about the problem as scientists and how that can help lead us to understand the biology of FSH and then to design what we hope will be effective therapies in the future. So I'm going to start with, is that working? No. Arrow buttons aren't working. Yeah.
Then there's an, yeah, I don't know. There we go.
Okay, I guess I'll
See that, yeah.
Okay. Okay, yeah, so I'm going to start with this cartoon, and I think this is something that a lot of people in the audience might have seen before. It's an illustration of what we refer to as the central dogma of biology. And the reason I want to put this up there is because even at this very fundamental level, we think about things differently than you're probably used to thinking about things if you follow science through the news or through newspapers or magazines and such. So you're probably used to hearing people in a magazine say, oh, we found the gene for something, or the gene that does this. So an example is, let's say, okay, there's a gene that makes your hair red. But the way we think about things is we don't really think about genes in that way, because genes don't actually do things the way that the popular media displays genes doing things. Really, all genes kind of do is they all kind of do the same thing. What they all do is they all just send a message to the cell that tells it to do something else. So the message is sent in the form of, try it this way. So the message is sent in the form of an RNA molecule. And the RNA molecule will go to a different part of the cell, and it directs the cell to make a protein. And proteins are really where the action is. So proteins are what actually does everything. So when someone says, oh, there's a gene for red hair, what that really means is that there's a gene at the DNA level that directs the cell to make a message, that tells the cell to make a protein, and then the protein will go and synthesize the red pigment that makes your hair red. So why that is relevant to FSH is that we're all kind of used to talking about the DUX4 gene, right? And Larry talked about this earlier. The DUX4 gene, everyone has it. You're supposed to have it. But most of the time, it's switched off. So there's no message being made, and there's no DUX4 protein being made in your muscles. Because in the cases of FSH, the gene becomes switched on, we get a message made, and then we get the DUX4 protein. And the reason that that's a problem is because the DUX4 protein does a lot of different things, but they all end up with the same thing, is that it's toxic to the cell. So when I think about how I want to understand FSH in a way that I can then develop drugs to deal with it, is to understand that the problem really happens at the protein level. So I want to understand as much about the protein as I can in order to then go ahead and try to develop drugs that will target the protein. Okay. So this is just kind of an illustration of a lot of the research that's been done about DUX4 and what we know about what it actually does to a cell. Now, obviously, this is very busy. I don't expect anyone to read through it and kind of understand everything that's on there. What I wanted to do is just use it to point out that there's been a lot of work that's identified that when DUX4 is expressed, there's a lot of different processes in the cell that all become disrupted simultaneously. And these probably all contribute to pathology in some degree or another. But what's been kind of missing from this chart, at least when I started, was that we don't really know how DUX4 causes all these problems. We know that they all happen. We know that the cell has a number of different pathologies that are all very characteristic of DUX4 expression. We don't necessarily know what DUX4 is mechanistically doing that's causing all of these issues to happen. So when I started my project here at UMass in Charlie Emerson's lab, I wanted to start figuring out exactly what DUX4 is doing that causes all these problems to happen. So now in order to do that, I have to tell you just a little bit more about the way that proteins work. So up until this point, I've been talking about DUX4 as an isolated protein, kind of independent of everything else in the cell. But that's really not the way that proteins tend to work. Usually proteins have binding partners, and that the two of these binding partners will cooperate together to do with it. But even this is still kind of a little bit too simplistic, because sometimes proteins combine with many partners. and then the partners combine with even more partners. So in reality, when we talk about a single protein, usually it's part of a much larger complex that all kind of fit together as like a pieces of a machine. And then the machine can then go and accomplish one particular thing that it does. Then it gets even more complicated than that, because usually these individual machines are all part of a larger network. So the entire network will all be responsible for carrying out one larger goal. So you can kind of think about this, a good analogy is to think of it as like different people working along an assembly line. Everybody has one particular thing that they do, and then you do one thing after another, and at the end you have the finished product. So these protein networks are really the relevant unit of what we have to think about when we think about what's going on with DUX4. Now, obviously, this is really complicated and it's very hard to understand, right? But the complications and the difficulties are also opportunities. Because what these big complicated networks mean is that if we can figure out exactly what network DUX4 acts within, and we can understand that network, then that means that every point along the network is a potential place for us to intervene and develop therapeutics. And then on top of that, if we understand the network, we can actually know where the best places to develop therapeutics are. And that's because networks tend to organize around nodes. So if you look here at this part of the graph, there's a node here. You can see there's a lot of different proteins that all interact with one particular protein. And that's kind of the center of a big part of the graph. So we can use these networks to identify multiple places where we can develop drugs, for. So when I first started here, what I wanted to do was figure out how DUX4 interacts with other proteins, how those proteins fit into a network, and then to try to develop therapeutics around that. So there's a lot of different approaches you can take. Larry already alluded to one earlier. There's genetic approaches you can take, but I wanted to take a protein-centric approach because it fits my background better. It's what I know, it's what I'm comfortable working with the best. So the approach that I decided to take was based on identifying the proteins that physically interact with DUX4. So the way we do that is, if you imagine a cell, and it's kind of full of all different proteins. There's, I've put a few dozen up here, but there's actually thousands of different proteins in a cell. What we can do is we can take advantage of antibodies. So that's what these guys are. So you're probably familiar with antibodies through talking about the immune system at various levels. We're not using the immune system per se, we're just taking the antibody from it. And the reason we do that is because what antibodies do is they will stick physically. They'll bind together like a magnet to one particular protein out of all the proteins that are present in a cell. And the reasons that that's useful is because then we can grab the antibody and pull it out away from the rest of the cell. And all the proteins we're not interested in stay behind. And the ones we are interested in, the ones that physically attach to DUX4, will all come out with it. Then what we can do is we can take this sample that we've just made and we can feed that into a machine called a mass spectrometer. So I could very easily have someone from our mass spectrometer facility come and spend all day explaining to you what this machine is and how it works. I'm not going to do that. I'm going to say what this machine does, as far as what I care about what the machine does, is it puts names on the proteins that come out. So once we run them through the mass spec and we can see, oh, here's the names of a few different proteins that we know DUX4 interacts with, we can then go into the literature, figure out what's been learned about all of these proteins already, and then we can start to figure out what network DUX4 fits into. So when I did this, the most interesting protein that I came up with was this one called C1QBP. And the reason for that is, if you look at what DUX4 does to the cell, so this is the same chart I put up earlier, and then you read the literature about what C1QBP is known to do, its functions overlap with this chart almost perfectly. So it was a really good candidate for a protein that might be mediating DUX4 effects as part of its network. And what was really convenient about C1QBP in particular is that it's already been placed within a network. So it's called the hyaluron network. And I can't pronounce that, so I'm going to call it HA for the rest of the talk. And I just advanced this too fast. So the HA network has actually been pretty well established. So what is it? So the HA molecule itself, that's represented here by this squiggly line, it's a molecule that cells make, and it does kind of two different things. it forms kind of what we call the extracellular matrix, or part of the extracellular matrix. And that's just a physical medium that cells use to attach to when you form a whole body. But it's also a signaling molecule. So it's something that's present that cells can detect. And then there's this whole big protein apparatus that detects whether the HA is there and then sends signals to the cell that it needs to do something in particular. So in this case, it might be to proliferate, it might be to move, whatever the case is. It directs the cell to execute a particular function. And as it turns out, C1QBP is kind of right at the base of this function. So the apparatus detects HA, sends signals, and that directs C1QBP to execute whatever it is that it needs to execute. So once we knew that this network was what we were working with, then we could start asking questions about what happens if we manipulate the network and what's the network doing in cells normally. So to start to look at that, we just did some pretty basic microscopy experiments. So what you're looking at here, Larry basically already showed this. This is what happens when we grow healthy muscle cells in cultures in our laboratory. So they form these very thick layers, and they look, this is basically what healthy cells look like. Now we can also put the cells through a particular procedure that will cause them to glow green if the HA network is being activated. So now what happens when we look at healthy cells is there's really not anything there. So I think you probably can't even see anything at all. There's a little bit of signal if you look at the screen, but basically the takeaway message here is in healthy muscle cells, there's not a lot of HA being activated. But then if you look at cells that are expressing DUX4, what you can see is, first of all, the cells themselves look very different, right? There's a lot less of them because they're dying and floating away. And then even the ones that are there, they look very different from what we see in healthy cells. But if we look at the HA, what we see is now it's being flipped on, right? So there's quite a bit of HA signaling going on in cells that are expressing DUX4. So that can lead us to ask, well, what happens if we have cells that are expressing DUX4 and we stop them from expressing HA? And it turns out that there was already a very well-known, well-established compound that can inhibit HA from being activated. So then we can see if you give that to cells that are expressing DUX4, the cells don't die. And they also look quite a bit more like healthy cells than they do like the sick cells. And you can see at the bottom here, we've stopped HA from being activated. So this is a potentially really exciting result, right? Because now this HA inhibitor is something that we can develop into a drug in the future. And we're putting together plans now. In a few weeks, I'm going to go visit some of our colleagues in Ohio, and we're going to start learning how to test this compound as a potential drug using the mouse model that Larry talked about earlier, that the Harper lab in Ohio has developed. That's not what this is supposed to say. Okay. So this is something that we're potentially really excited about, because this is a very encouraging type of drug. But we don't want to stop there, because as I'm sure everyone here knows, very few drugs that you try to get through actually wind up being the drug that's useful in the end, right? There's always a million candidates for every one thing that becomes an actual drug. So we don't want to go into testing with one drug. We want to go into testing with several drugs. And to make that possible, we can go back and look at the HA network. And as it turns out, part of the HA network down at the bottom here are these, what we refer to as signal transduction pathways. Now, these are probably the best understood pathways, maybe in all of biology. That's because they're very fundamental to biology, and they're also tend to be dysregulated in cancers. So these have obviously been just an intense amount of study has been focused on these signal transduction pathways. So the benefit to the FSH community is that there's already just a whole host of drugs that are known that affect these pathways. So that means we have the potential to take these drugs that are already being used for cancer applications and repurpose them for FSHD. So I've only just started working with these. We formed a collaboration with Genea Biocells, and they provided us with a number of different compounds that we could screen through. And so this is the only graph I'm going to put up, I promise. So I've just started screening through using an assay that detects how cells respond to these different drugs. So what you're looking at here is it's an assay that tells you how healthy the cells are. So in healthy cells that we happen to put DUX4 in, we're just defining that as 100. In cells that you've activated DUX4 in, it's about half, right? They're about half as healthy as our control cells. And then we can take cells that are expressing DUX4, also expose them to the screening compounds that we got from Genea. And you can see that several of them at higher doses are looking quite a bit healthier than the untreated cells. So these are potential drugs that we could also develop more in the future. And I say this is very preliminary, like we've just started doing this. So even things like the dosage that we've used here, these were barely more than a guess. So there's quite a bit of optimization that we can do here that can hopefully be able to bring these bars much closer to the top. And then once we do that, then we can also take those with us to the Ohio group and start testing them in their systems and also in the xenograft systems that we have going on here. Okay, so I'm going to hand this off now to Caitlin, and she's going to talk more about how we actually do those types of testings. So I just want to stop and just say thanks to everybody who's contributed to the project. There's been a lot of different people have given me ideas and also reagents. Larry's been very helpful, the core facilities we have here. But also I just really want to highlight, it's really the FSH Society that has pushed this through with funding and connecting me with colleagues. And if you wonder where the research dollars go, it's to this type of project that they've been funding for me. And then also to the Wellstone Center that has also picked up a lot of the funding slack. Okay, and if there's any other questions, I can take those.